Okay, in part 1 of Module 18, we already talked about characteristics of visible light. You know that has to do with wavelengths, both amplitude and length, shortness, all of that. And we also described how the eye processes visual information in the brain. But we didn't talk about theories that help us understand color vision. So that's the last part, and this will not be 15 minutes like the other was, okay? So I want you to stare at the green part. I probably won't wait 30, actually I'll wait 30 seconds. So you stare at the middle of the flag for 30 seconds. You really have to focus and just keep your eye on the middle of the flag, the middle of the green intersection. Don't let your eyes dart around. Try not to blink. It's better if you don't blink. 10 more seconds, stare intently. Then pay attention to what you see carefully now. So if you're looking carefully at the screen, it should be like you see pinkish where the green used to be and blue on the outside edges, probably framed by white. So there should be a red cross. We're going to talk about why that happens, or what's the theory around that. So we talk as though objects actually possess color. Tomato is red. Perhaps you've pondered the old question, if a tree falls in the forest and no one hears it, does it make any sound? We can say the same thing about color. If no one actually sees the tomato, is it still actually red? The answer is no. First, the tomato is actually everything but red because it rejects or reflects the long wavelengths of red, which means they bounce back to you and you see them. Second, the tomato's color is only our mental construct. As Isaac Newton noted, light rays are not colored. Color, like all aspects of vision, resides not in the object, but in our brain. One of the vision's most basic and intriguing mysteries is how we see the world in color. How from light energy striking the retina does the brain manufacture our experience as color? And a multitude of colors. Our different threshold for colors is so low that we can discriminate more than 1 million different color variations. At least most of us can. For about 1%, about 1 person in 50, vision is color deficient, and that person is usually male, because the defect is genetically sex-linked. So why are some people color deficient or vision deficient? To answer that question, we need to understand a little about how normal vision works. Modern detective work on this mystery began in the 19th century when Hermann von Hemholtz built on the insights of an English physicist, Thomas Young. Knowing that any color can be created by combining the light waves of 3 primary colors, red, blue, and green, Young and von Helmholtz inferred that the eye must have 3 corresponding types of color receptors. Years later, researchers measured the response of various cones to different color stimuli and confirmed what is now called the Young-Helmholtz trichromatic 3-color theory, which implies that the receptors do their color magic in teams of 3. Indeed, the retina has 3 types of color receptors, each especially sensitive to one of 3 colors. And of those colors, they are red, green, and blue. When we stimulate combinations of those cones, we see other colors. For example, there are no receptors especially sensitive to yellow, for example. When we see yellow, we are mixing red and green light, which stimulates both red-sensitive and green-sensitive cones. Most people with color-deficient vision are not actually colorblind. They simply lack functioning red or green-sensitive cones, or sometimes some of both. Their vision perhaps unknown to them because their lifelong vision seems normal. It is called either monochromatic, 1 color, or dichromatic, 2 color, instead of trichromatic, making it impossible to distinguish between red and green. Usually there's 1 person in class that is colorblind. Dogs, too, lack receptors for the wavelengths of red, giving them only limited dichromatic color vision. But how is it that people blind to red and green can still often see yellow, and why does yellow appear to be the pure color and not a mixture of red and green the way that purple is red and blue? Edwin Herring soon noted trichromatic theory leaves some parts of the color vision mystery unsolved. Before we move on, If you are dichromatic or monochromatic, you might struggle to see the image on the right. So depending upon which of your cones are deficient, most of us can see that there's a 57 clearly in this image. But for those people with monochromatic or dichromatic vision, it's possible that they don't see that very clearly or at all. So Herring, a physiologist, found a clue in after images. Staring at a green square for a while and then look at a white sheet of paper and you'll see red, green's opponent color, opposite color. Stare at a yellow square and the opponent color of blue will usually appear on a white paper. To experience that, we tried this at the very beginning. We are going to look at the flag again now that you kind of know what you're looking for. Herrick surmised that there must be two additional color processes, one responsible for red versus green perception and one for blue versus yellow. Indeed, a century later, researchers also confirmed Herring's opponent process theory. Let's take a look at that. Three sets of opponent retinal processes, red, green, yellow, blue, and white, black, enable color vision. In the retina and in the thalamus, where impulses from the retina are relayed en route to the visual cortex, some neurons are turned on by red, but turned off by green. Others are turned on by green, but off by red. Like red and green marble sent down kind of a narrow tube, red and green messages cannot both travel at once, so we do not experience a reddish green. Red and green are thus opponents. But red and blue travel in separate channels, so we can see like reddish blue, like a magenta color. So how do we explain after images, such as the flag demo? By staring at green, we tire our eyes green response, and when we then stare at white, which contains all colors, including red, only the red part of the green-red pairing is going to fire normally. The present solution to the mystery of color vision is therefore roughly this. Color processing occurs in two stages. The retina's red, green, and blue cones respond in varying degrees to different color stimuli. As the young Hemholtz trichromatic theory suggests, their signals are then processed by the nervous system's opponent process cells, as Herrick's theory proposes. Okay, so let's try that one more time. So this is just a demo of Herring's theory. Okay, so one more time you're going to stare at the middle of this flag. Stare at it hard. "15 more seconds. Stare intently at the middle. And now pay attention to what colors you seem to see. So I see blue, white rimming the red in the center, and then it fades away fairly quickly. Okay, one more after image experiment and we'll be done. Okay, another perception experiment. I'm going to push the button and this spiral is going to start to turn. And I want you to guess, you have some options on the right here, after you're done staring at this intently for about a minute, what's going to happen to your vision? So do you think the photo that I show you is going to rotate counterclockwise? Do you think the photo is going to rotate clockwise? Do you think the photo is going to pop up and then zoom toward you? Or will it appear and then shrink away from you? What do you think? Pick one. And then stare at the center. It's going to seem like a long time, all right? So here we go. Stare at the center. Try not to blink. Stare at the center. "30 seconds in, keep staring at the center. "15 seconds to go. Keep staring at the center. Stare hard, 5 more seconds. Okay, if you blink and kind of readjust yourself, you can see that this photo is just a regular photo. It's not moving or anything, but for most people, that photo for about 2 or 3 seconds appears as if it's kind of coming at you, zooming and getting larger. It just goes to show that your perception is manipulable, and depending upon movement and color saturation that's around you, things can either move or the colors that you see can change based upon your perception. So your brain's perception is definitely a flexible thing, and it's unique from one person to the next.